Welcome, everybody. I'm Terri Conneran with KRAS Kickers. I am a patient and a founder of the organization. And I can't just tell you how much I'm just happy to be here. It's like we haven't had an opportunity to really focus in detail on pancreatic cancer. And so what a better way to really just kind of get all jacked up about it with Dr. Andrew Aguirre. We read to you a brief bio on him because I can't possibly cover all of it, but we're just going to hit the highlights. Andrew Aguirre is an MD, PhD, so that's double trouble, I believe. Medical oncologist at Dana-Farber Cancer Institute. He's an associate professor of medicine at Harvard Medical School, an institute member at the Broad Cancer Institute of Harvard and MIT, and associate director for the Hale Family Center for Pancreatic Cancer, co-director of the Center for RAS Therapeutics at Dana-Farber. Dr. Aguirre leads a basic and translational cancer research lab that studies pancreatic cancer biology at its source, behind the bench, and then RAS signaling with the goal of developing new therapeutic strategies for patients. And it's the first time we've actually had him come on, even though we've had a couple other people from Dana-Farber. Welcome. Thank you so much for joining me today, Dr. Aguirre.
Oh, it's my pleasure. Thanks for having me, and thank you to all of you listening for your interest to talk a little bit today, and honestly for your ongoing advocacy and fight against cancer. So appreciate you all being here.
Well, truthfully, most of us didn't want to come into the fight to begin with. And so we're just here, like, just hoping that we can come up with some plans, some strategies, and insights as far as what's going. And so I'm going to kind of like let you kick off. I mean, I can hit you with 1,000 questions off the top of my head. But I'm not going to do that. And just to kind of for clarity, if you're okay, I'm going to let you talk and do a presentation of sorts, and then we can kind of like engage back and forth and go through questions as is.
Sounds good. Yes, I'm happy to take any questions as well. But so as Terri mentioned, I'm a medical oncologist, and I run a research lab focused on pancreatic cancer primarily. I've been studying pancreas cancer for about 25 years now, ever since I was a first-year graduate student and medical student. And if there's one thing I've come to learn early on is that it's a difficult disease and it's a challenging disease, and it's been challenging to study over the years. Not a lot of great model systems originally, not a lot of funding for the research in the earliest days. But I would say over the last couple of decades, really, the community has just built a fantastic army of laboratory investigators, clinical investigators, really working to, turn the tide on this disease and really change the course of clinical care and really make a tremendous impact for patients. And we've all been working very hard on this with, advances in chemotherapy and surgery and radiation, but none more important or timely or potentially impactful than the recent advances in small molecule inhibitors that can inhibit the KRAS protein and can block this major driver of pancreas cancer. And it's important to note that almost all pancreatic cancers are driven by KRAS. In fact, 95% of pancreatic cancers have a KRAS mutation. Those mutations occur in the earliest stages of the disease, including in what are termed pre-invasive lesions or early cancers, pre-cancers that occur in the pancreas. Those are driven by RAS, and those can progress and ultimately become advanced cancers or cancers in the pancreas that then can spread outside the pancreas. So we know that KRAS has been really the holy grail target for pancreas cancers and really the elephant in the room. We can develop drugs against many other targets or opportunities, but really blocking KRAS is really central to the mission of developing better therapies for pancreas cancer. And so really with the advent of many different molecules, in fact, now there are over 60 distinct molecules in clinical trials, 60 different molecules from different companies, and really not 60 trials, but 60 different molecules. So there's just an avalanche of new therapies that we're testing in patients, looking at in the lab actively and trying to understand how they work, when they work, if they don't work, why do they not work, and how do cancers become resistant to those drugs. And importantly, what do we add to those drugs to make them work even better, to make cancers go away and stay away for as long as possible. And so my laboratory has studied RAS inhibitors for a long time now, and really initially in small subsets of pancreas cancer that have mutations in a particular mutant form of a KRAS, a codon 12 mutation, a G12C mutation. There's a whole fleet of inhibitors that work well in lung cancer and colorectal cancer, and also have evidence of early signs of efficacy in pancreatic cancer. But perhaps even more impactful have been small molecule inhibitors that also block the many other forms of KRAS that occur in tumors, including other mutations at the codon 12 position or codon 61, these various spots in the KRAS protein that can cause that protein to be overactive and drive cancer. So now we have RAS inhibitors for just about every flavor of KRAS mutation in pancreas cancer. And that's opening up tremendous opportunity to bring these drugs to patients and to eventually improve the quality and duration of patients' lives with pancreas cancer. And so my work and interests in the lab and in the clinic really center around bringing these new drugs to patients and understanding how to make them work best. And the work that I do is highly collaborative, and we work very closely with other members of Dana-Farber, but more broadly, other members throughout the country and throughout the world to try to work together and not to duplicate efforts, but to build on the efforts that everyone is doing. And I've known Terri now for quite a while because Terri's at all of the same meetings and talking about all of the same concepts that we are, and really helping to spread the news and the mission, and to really spark collaboration and dialogue, and really encouraging people to work together and to discuss findings early and often, and most importantly, to always keep the patient first and the patient centered around what we do. So that we translate those advances as quickly as possible to patients and hopefully enable them to have the most impact for patients. So that's a little bit about what we do and what my lab works on, and just sort of my overall interest in the field. I'm happy to take any questions, or I'm happy to expound further on in any number of dimensions.
Oh, I'll hit you up good, and thanks for kicking it off like that. All right, so let's start with like the basic, and then we'll kind of roll into it a little bit more, okay? Because we have a huge wide variety of people that come on and join, from somebody who might have just been diagnosed with a cancer to somebody that is like not only diagnosed, I mean, they're very, very educated about the cancer, or just even researchers and stuff. So we have a wide scope of audience, and that's why we're real selective about like who gets to come in and speak. And so like, I mean, it's like you're one of the pick of the litter. So
Appreciate that opportunity.
but I'm saying that merely because I need you to understand who we're going to be talking to. So starting with that, okay. So let's explain, just kind of simple terms, what an inhibitor is, what that means, and why I would want to do it, and how that might be a little bit different. So let's start brass tacks, and then we'll build on that.
Great. Yes. No, important to know. So cancers are diseases of abnormal genes that happen in the body, and they changes in genes can lead to abnormal growth and proliferation, and increases in size of these number of cells and the tumors themselves. So understanding that, a cancer occurs because there are changes in the cells that lead to dysregulated growth and survival of those cancer cells. The KRAS gene produces a protein, the KRAS protein, and there are in those abnormal KRAS proteins, a switch is flipped that is a mutation or a change in that protein that leads to it being it locked in the on state. So in the go, 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 it's like having the gas pedal pushed down in a cancer cell and stuck down, that cancer cell's being told to grow, grow, grow. So these inhibitors are small molecules or small drugs that you can take typically in a pill form. And those drugs get into the cells, all of the cells of the body, and go spread throughout the body. But then they home in on the cancer cells as well, and they can block the function of that abnormal protein, the KRAS protein. They can prevent it from being stuck in the on state and the gas pedal forced down. It can reverse that, and that they can block that abnormal growth signal from the KRAS protein, or they, in some cases, can reverse it and make it act more normal. So these inhibitors are really drugs that you take by mouth. So similar like an ibuprofen or a Tylenol or a statin or something that you take by prescription, but you take that by mouth, that gets into the cancer cells, blocks the function of this abnormal RAS protein.
Okay, so I'm going to repeat back, make sure we're on track, and then we'll kind of move forward. So you're saying that KRAS, you need it in your body to begin, right? And then something triggers it, and then it starts making the wrong protein there, and it keeps multiplying that. And then when we're looking at an inhibitor, we're looking at that thing to wedge in there to stop it from making that wrong protein, right?
Yeah, that's right. And I think that's a good way to think about drugs in general. In these building blocks, proteins in cells, drugs are designed to fit like pieces of a puzzle. That there may be a triangle in the RAS protein, and you develop a drug that's a triangle that fits right into that piece, and that blocks the function of that protein. So it's a lock and key or a pieces of a puzzle, but you think about drugs that way, that you build them to fit a specific protein, and in many cases, that abnormal form of the protein that's in cells. If you can develop drugs that specifically block that abnormal protein and not the normal protein, because as you said, everyone has KRAS in all of their cells, and it has a very important normal functions. It's really a regulator of when your cells grow and divide and how they do that. When it's stuck in the on position, that's bad. So we need to come in and block it with a drug.
Okay, all right, because that's one of the things we get all the time. Okay, and then the next part of that is everybody sees the G12C inhibitors that are on the market and the FDA, and it's like, well, it doesn't seem that different, a C to a D or an A or whatever, but it's not just a letter. It represents an entirely different door in an entirely different, like kind of almost neighborhood. So if you kind of explain that, we'll kind of keep going.
Yeah, that's very important, especially for patients or clinicians or people on the call who are particularly interested in or experiencing or struggling with pancreatic cancer, because KRAS G12C mutations, or that flavor of RAS, is actually quite uncommon in pancreatic cancer. In fact, as Terri indicated, there are different versions of RAS that are the major drivers of pancreatic cancer, and those are G12D or V or R for the most part. And it's critically important that those G12C inhibitors, those drugs that block the KRAS G12C protein, don't typically work on G12D or V or R mutations, or the mutations most commonly observed in pancreatic cancer. And it's very important that specificity for the G12C protein is what makes those drugs so good for the cancers that have KRAS G12C mutations. Lung cancers, colorectal cancers, a small fraction of pancreatic cancers, because that specificity is what you want. You want it to block the abnormal protein, and you want it to leave the normal RAS protein alone, because that's important for function and for all of us to not have major side effects from these drugs. So the real opportunity and challenge has been to develop RAS inhibitors. The G12C inhibitors came first. That was kind of that was the dam that broke and showed us, wow, we could target KRAS and we could block these proteins, and that could benefit patients. The opportunity challenge then came to develop drugs that block these other flavors of RAS as well. And what we've seen now in the last 5 years or so is a whole host of other drugs that can block G12D, VR, other mutant forms of RAS that we see most commonly in pancreatic cancer. And as Terri, as you suggested, getting the right lock and key combination of the mutation and the mutant form and the drug is critical. It's critical to be able to block that protein effectively. And it's critical to leave the normal proteins alone so that we can balance anti-tumor effects, blocking the tumor, with leaving the normal cells alone and thus preventing side effects and toxicity.
Okay, so it underscores the level of importance of biomarker testing in cancer.
Definitely. And and by biomarker testing, I think what you mean is we need to find markers that suggest a patient is going to benefit from a specific drug. And those markers most often are genetic markers that you take a biopsy of the patient's tumor and you do what's called DNA sequencing, where you sequence all of the genes in that tumor and you home in on the ones that are different from normal cells. And those different DNA mutations, most notably a KRAS alteration, and a mutation in the KRAS gene that leads to an abnormal protein that drives the growth of those cells, we have to find those mutations because those mutations require specific drugs. And if we can match the right mutation with the right drug, the right patient with the right drug, that has the most likelihood of benefiting patients. So biomarker testing is critically important. And that's become a standard process and a standard way of thinking in oncology, where we have to match the right patient to the right drug. But it all begins with understanding what drives that patient's tumor. And then how do we go after it and eliminate it?
Well, I know it's really standard at Dana-Farber and a lot of the big institutions, but it's not necessarily standard practice at a lot of the other ones. And so I think that's part of the issues. And okay, I'm going to tell a tale on myself here. It's like when I found out that I have KRAS G12D and it's so common in pancreatic cancer, it blew my mind how it was the same cause, the same problem. And it was like, I had this commonality with something that was so different than what I had. And so it scored, it scared me from the standpoint of it's like cancer is many different types of cancers, but it's all boiling down to one molecular area. That was really poorly articulated on my part. But
No, I think that's very important. And you make a really important point. And I think what you said is true, that these different forms of KRAS are different locks that require different keys, or different doors that need to be opened with different drugs. That said, they very similarly do the same thing. There are different ways to turn that protein on, to push that gas pedal down. And if we can develop drugs that by and large block the end effects of that could be very powerful. And so there are drugs, we're talking about drugs like G12C drugs or G12D drugs. Those exist. They're being tested in the clinic currently in pancreatic cancers and many other patients with other types of tumors. But there are also drugs that have been developed that block all RAS proteins or all different mutant flavors of RAS. And that's very important as well, because it's not always possible to develop a specific key for every lock. But if you had a master key that worked for all locks, that could have really important benefits. It may have additional side effects in some cases if you're also targeting the normal RAS protein. But what we know is that these drugs are much better tolerated than we had expected. There's still side effects that for some of these master keys, that the drugs that block all RAS proteins, but those are being actively used in patients currently. And we're learning how to mitigate those toxicities and to balance them. And those drugs may be good enough one day to not require even understanding what type of RAS mutation you have, but just knowing, is it a RAS mutated tumor or not, and they may work for all different types of RAS mutations. So something on the horizon that some of these drugs may be applicable even in areas outside where it's easy to get a biomarker test, but you can look beyond that into the rural communities or outside of the United States or internationally in areas where it's not so easy to get these testing done. So I'm optimistic that down the line, these drugs will be accessible to a broad patient population. We just have to run the right trials with the right biomarkers and understand how these drugs work in patients to ultimately one day get them to as many people as possible.
Okay. That was a nice recovery of what I was saying. You keep talking. Let's drill down a little bit into the difference between KRAS and RAS, because that's kind of one of the things that's getting lost in the nuance. And I have about 1 billion questions that are coming in, and I already have about like what this new, the other ways of on and pan and this sort of stuff that we're gonna be talking about. So I'm trying to establish like a baseline first so we can kind of go bouncing into that.
Yeah, that's a good question. And it does get a little bit nuanced into what different genes are. But RAS proteins, you can think of it as 3 major forms of RAS in a cancer cell. KRAS, which is by far the most common driver of the RAS proteins and really one of the most common drivers of cancer in general, of all genes. KRAS, by far the most commonly mutated or altered in cancers. HRAS, which is another type of RAS protein, and NRAS. And these 3 genes, which produce proteins, are what I like to think of as like kind of look-alike genes, that they're very similar. It actually, in sequence identity, in the structure of those proteins, they're way more similar than dissimilar, than they're not alike. So those proteins are very highly identical, but there are small differences in those proteins. There are 3 distinct proteins that have overlapping but sometimes distinct purposes in the cell. So there are 3 forms of RAS, and within each of those forms of RAS, there are many different mutations that can occur in those RAS proteins. So when we talk about RAS inhibitors or KRAS inhibitors, we have to think about which of those 3 forms of RAS are those drugs hitting, and which of the mutations in those forms of RAS do those drugs work against. So there are what I would term mutant selective inhibitors that block only G12C or D or V. And then there are pan-KRAS inhibitors or KRAS inhibitors that block only KRAS, but they block many of the different mutations seen in KRAS. And then there are what I would say would be pan-RAS inhibitors, or that term pan means kind of all of them, all across them. So they block all RAS proteins, KRAS, HRAS, and NRAS, including many, if not all, of the mutant variants of those proteins. So there's enormous complexity to these RAS inhibitors or KRAS inhibitors. And that's where really the insights that we've learned over the last several years about which of these drugs block which proteins, and saying that a tumor or a patient's tumor has a KRAS mutation is no longer enough to really know which of those drugs should work. And that is a super important point and an important challenge, but a very good challenge to have, because it means that we're developing drugs against these different proteins and these different forms of RAS. And I'm very optimistic that one day in the not too distant future, we're going to have a RAS inhibitor for every variant of RAS. And it's just going to be a point about finding the right mutation and the right inhibitor for that patient and figuring out what to combine with it to make it work better.
Okay. That's a great explanation. Thank you. Okay. And so from there, all right, now let's talk about some of the actual treatments and drugs that are going out there. And one of the places I think needs to be started is we saw that there was a oh, what I should have written, I should have put it on my screen, with the RevMed drug. And it had breakthrough designation, and now it has this, I don't know, what are they calling this? This super fast tracked thing?
It's a voucher, yes.
Yes,
FDA
yes,
yeah.
okay. And I know that this hasn't happened before, but what is your take on what this is and how long it's going to be? Because, and what this means for patients and for doctors in cancer treatment. So elaborate on it. You're the expert. Please help us understand what this means and what this means from a real world perspective.
Yeah, this is an important point. So this drug that Terry's referring to is a drug called daraxonrasib, a complicated name, long name, but the drug's been shown to have good anti-tumor responses and good efficacy in patients in lung cancer and pancreatic cancer and even colorectal cancers. And the drug is now in 2 large phase 3 randomized controlled studies. So those are what that means is that they take patients and they want to test these drugs in patients in a very rigorous way so that they know that if you get the RAS inhibitor compared to what you would get as standard of care, current standard of care, does the RAS inhibitor work better than or as well as what the standard of care is? And so they run these large studies, well controlled across many different sites, to really answer that question. Is this drug safe and effective to give to patients? And so those large studies are ongoing to test these drugs in patients with lung and pancreas cancers. And what a breakthrough designation, what that means is that the FDA, the Food and Drug Administration in the United States, recognizes the potential of these drugs, is paying very close attention to them, and is going to try to expedite the review of that data when it comes out, and ultimately the decision about approval of those drugs to be used in a more broad spread manner. Meaning right now these drugs are only accessible in clinical trials. Eventually, where we and many patients struggling with these cancers would like to see it is we'd like to be able to write a prescription to get that from the pharmacy. These drugs are now accessible to patients, they're covered by insurance, and they can be prescribed by oncologists to be used in any patient that needs them. But that process of getting from promising drug in the lab to safe drug in patients to demonstrated effectiveness in patients needs to be a methodical, well-done process, because first and foremost need to be protective of patients and develop these drugs in the right way so that we understand if they have side effects or that they work in patients. We also need to develop the drugs in well-controlled studies so that we can say definitively, how do these drugs work and how does it compare to what the patient would get otherwise? And then the results of those studies really need to be reviewed by the Food and Drug Administration and many academic and industry experts to be able to agree that these drugs are beneficial to patients and get them to patients. And what the FDA is saying is that this is an important drug. We're paying close attention to it, and we're going to try to expedite this as much as possible.
So from a timeline perspective, what I'm hearing you say is that it looks like it's going well, and they're just going to move it to the top of the paperwork pile to make sure that it gets reviewed faster than
I think what they're saying is that the early data from the phase 1 clinical trial, the first in human studies that show how is it tolerated and what are the early signs of efficacy, were encouraging enough, and the need is significant enough that there are so many patients with tumors that have RAS mutations that this is an urgent need to address this. That need is so big that there's a priority designation on these drugs, because those drugs can address that urgent need. And so we really need to wait for the results of this phase 3 randomized controlled trial in pancreas cancer. Many patients have been enrolled, many patients are still on the study, and the jury's still out, and we're waiting on the results of those of that study. And so and when that reads out depends on when the patients were enrolled and how long they stay on the drug, and when do they have enough patients reached the efficacy markers, efficacy benchmarks, the effectiveness benchmarks on that study to be able to stop the study and look at it and compare the arms of the trial and make a decision. But we're not talking 5 years, we're not talking 10 years, we're talking, in the next couple of years that these studies are going to mature and read out and hopefully advance to approvals for patients.
Well, you know what I have to say to that. That's not, a couple of years isn't fast enough.
Yeah, I think we all feel that way. Patients feel that way. The companies feel that way. The companies want to see these drugs developed for patients. But what we've learned in drug development and oncology is you have to do things the right way to ultimately benefit the most patients in the long run, and to get it right with these drugs. Doing that ensures that these drugs are going to be accessible to as many patients as possible. But we all agree we need to move at lightning speed with this and with these drugs, and all drugs, all RAS inhibitors, additions to RAS inhibitors. There's no time to waste. And even as quickly as we can move, it's still not quick enough. We really feel an urgent need to develop these better therapies for patients. And we feel it every day and all the time. So we're always looking for ways to accelerate that, for ways to find the next new insight in the lab that's going to give us a jump start on what we should do next in patients, and ultimately to work with patients. The patients are allies and partners in these clinical trials, and they're every bit an important part of doing these trials the right way, and understanding how these drugs work, and learning from them along the way.
Okay, so I wave a magic wand and tomorrow the FDA says, okay, this diroximib is approved. Okay, ta-da, you're welcome. What does that mean? From a practical standpoint, what does that mean?
Well, that means that in the study where this is being tested in pancreas cancer, for instance, are in patients who have had prior chemotherapy, advanced pancreatic cancer that spread beyond the pancreas, patients that have had prior chemotherapy and their tumor has grown on prior chemotherapy. An FDA approval of the drug on the basis of those trial results would mean that those patients would be eligible to get daraxonrasib after their tumor has grown on chemotherapy. And that growth on chemotherapy, that drug would then come in what we'd say the second line setting and be usable in that setting. But more generally speaking, what it means is that many more patients are going to get access to this drug, and an approval in that setting, especially when there's such an urgent need for new therapies for pancreas cancer, is going to mean that those drugs should be covered by insurance plans, and they're going to be broadly applicable. And it's going to be a drug that community oncologists could give, or oncologists in academic medical centers could give. And we're going to learn a lot more about the drug very quickly because many more patients are going to get the drug. And I think it's going to generally accelerate our understanding of how these drugs work and how they benefit patients.
Okay, so what I'm hearing you say is that once the drug is approved, it doesn't mean you just go into the local pharmacy and collect this medication. You have to still qualify to have the treatment. You need to have pancreatic cancer. You have to have already had chemotherapy and still had progression beyond just your pancreas. And then that would be who would qualify to get that treatment.
Yes, it still needs to be prescribed by an oncologist. There are indications for the drug upon an approval, and the indication or the certain scenario or context for any given patient has to match that approval. And that where it's being tested right now is in patients with advanced pancreatic cancer that's spread beyond the pancreas and who have received prior chemotherapy. But what that also means is that there will be other opportunities. That drug will be tested in the first line setting before patients receive any chemotherapy. And there are already plans underway to test that. There have been some studies that have looked at that, and there will be larger studies that will look at that. Or in patients who present to their doctor with pancreas cancer that's localized in the pancreas, has not yet spread elsewhere, but we know that we need to treat those cancers with systemic treatments, with chemotherapy or pills or drugs that block the cancer, because we know that we need to reduce the risk of recurrence. So use of these drugs in the advanced setting will also help set the stage for us to test them earlier in earlier lines of therapy, or in localized pancreas cancer, using it before surgery or after surgery to try to minimize the risk of recurrence and ultimately maximize cure rates. And so that's also on the not too distant horizon, is how do we take these drugs, which we think are going to be effective in the advanced setting, move them earlier and reach even more patients.
Okay, but it's still only in pancreatic cancer. It's not going to be, I mean, that's what the approval would be for, is not in this
Yeah,
particular
well,
set of data. It doesn't apply to the other cancer types, is I guess where I was going.
yeah, but there are studies ongoing in those other cancer types as well, and not just with daraxonrasib, with many different drugs to try to lung cancer, pancreas cancer, colorectal cancer, other tumor types, gastroesophageal cancers, cholangiocarcinomas, tumors that have RAS mutations. RAS mutations drive many cancers, including many beyond pancreas, colon, and lung. So there are studies ongoing in all of these diseases with the goal to advance these therapies ultimately to patients. But the data in support of using those drugs in those tumor types needs to be there to be able to pursue the approval to use it more broadly outside of clinical trials.
Science goes slowly.
Yeah, and sometimes science moves quicker than we can get things through the regulatory process in clinical trials. But that's what all of us do every day, is come to the hospital with the mission to advance science and science to patients. And it's a good fight to keep fighting, because I think we're on the cusp of moving much more quickly than we've ever been able to do so before, because there's been a slow buildup. We didn't have drugs that blocked RAS for 40 years. Now we have over 60 and counting, and many more in development in companies and in labs. And it's going to be an exponential pace, because these drugs are all in clinical trials, and we're going to very quickly in the next 5 years get to which of these drugs work well and best and have the best side effect profile, and how do we get these to patients. And I think we're going to see just an explosion of advances for patients in the coming years. And that sounds like that's easy to say, but 5 years ago we could not have said that. 10 years ago we could not have said that. There were not these drugs on the horizon. So the fact that we're looking at these times when we're starting to talk about targeted therapy in pancreatic cancer, or precision medicine, precise therapies that are engineered and designed for a particular patient's cancer, that's a relatively new thing for pancreas cancer. And so we're just incredibly excited about that, and about the potential benefit it's going to for current patients and future patients.
It gives me confidence to know that you start having a fighting chance as a result of all this.
Yeah, absolutely. And hope is everything in oncology. And I think when you have that, you do have a fighting chance.
Well, we have plenty of hope as patients, but we need the research and the speeding and the time to make it keep happening. So, okay, so with all that and that kind of going on, so let's start with the bigger chunk and kind of like roll down, because the KRAS G12D is so very common in pancreatic cancer. right? It's the most common mutation in pancreatic cancer, is my understanding. So if I have a KRAS G12D pancreatic cancer, what is the lines of therapy that I would be kind of like looking at was most logical compared to somebody that doesn't have that specifically?
So you mean if you had a G12D mutant pancreatic cancer?
Yeah.
Well, the current standards of care remain if it's advanced pancreatic cancer that's spread outside of the pancreas is chemotherapy. And there's multi-agent chemotherapy that you get at your oncologist office through intravenous infusions. And you come generally every 2 weeks back to the clinic to get those infusions. And you are followed with serial scans to check to see how the tumor is growing and blood work to see how the patient's doing and how the markers from the tumor. If it's localized pancreas cancer that's only in the pancreas, it's generally either resect the tumor and then get standard of care chemotherapy, what's called adjuvant chemotherapy, or get pre-surgical or neoadjuvant chemotherapy followed by surgery and sometimes radiation in there before surgery as well. And then ultimately you get more chemotherapy after surgery. So that's still really surgery, radiation, chemotherapy are the mainstays of pancreatic cancer care, irrespective of whether you have a G12D, V, R, no mutation in KRAS in your cancer. That's still the standard of care. Eventually, with the development of these drugs or G12D inhibitors or pan-RAS inhibitors, RAS inhibitors that block any form of RAS, eventually we need to figure out where to slot in a RAS inhibitor. Do we give that first as a therapy alone, a single therapy with a RAS inhibitor? Do we combine that with chemotherapy? And there's good data in the lab and even in patients and other paradigms in oncology and colorectal cancer or lung cancer that targeted therapy with chemotherapy. They make the combination works better than either alone. So there's good precedent that maybe combining RAS inhibitors in pancreas cancer with standard chemotherapy may actually work better than either agent alone. So figuring out RAS inhibitor, then chemotherapy, RAS inhibitor with chemotherapy, chemotherapy, then RAS inhibitor. That's what we need to figure out and run in clinical trials designed to answer a question and prove to ourselves what's the best way to give these. So that's where I see, the next, even beyond a first approval, the next questions, the questions that we need to continue to try to answer even now in advance of that approval, potential approval, would be how do we give these drugs in sequence or in combination? And ultimately, what do we combine with a RAS inhibitor to make those responses that we're seeing even more durable, to make the tumor shrink, stay shrunk, maybe even go away for a period of time or hopefully one day forever? What combinations of therapies do we need to give to make that happen?
Okay, and so that as it stands right now, there wouldn't be necessarily a change in treatment based on that. What you need to know though is that as you're moving through the standard of care situation, right? That's when do you look at a clinical, man, I'm off base today. Let's try this, let's try that again. When should I look at considering a clinical trial when it falls in the scope of my treatment cycle?
Yeah, good question. So what I was just describing is the current standard of care for patients with pancreatic cancer, which has not changed yet because of RAS inhibitors. The trials are being run to hopefully change that standard of care. And in that, the nearest term trial results that could change that standard of care, that would be in the case of patients who've received prior chemotherapy and their tumors growing on chemotherapy. Currently, there are many different clinical trials ongoing that are looking at KRAS inhibitors in that setting of after chemotherapy. That's the most common place they're being evaluated currently, but also trials looking at them prior to chemotherapy, before the patients received any treatment. So that would be the first line setting, or treatment naive cancers that haven't been treated with chemotherapy. But also looking at studies looking at, can we combine RAS inhibitors with other agents such as chemotherapy. Those trials are currently being run in early phase clinical trials to mainly to gauge the safety and early signs of effectiveness in patients. So there are studies out there looking at not just daraxonrasib from Revolution Medicines, but many other drugs, many other very good drugs in clinical trials, looking to see, do they work after chemotherapy, starting to look at, do they work before chemotherapy, and also how do we combine them with chemotherapy or other agents to make them work better.
Well, and then there's what's next. Like, because if the cancer comes back, okay, let's just from a practical standpoint, okay, I'm a cancer, I've had it come back 5 times, right? And so from a practical standpoint, you don't always go in and get the big win, right? And so you go in, you do the best you can, and then you go from first line to second line treatment. And then if it comes back, what's next? Those are still going to be ongoing questions and ongoing issues, right?
Yes, definitely. And we know that unfortunately the cancer often does come back. And it comes back and develops resistance to these RAS inhibitors, hopefully after many months of benefit, sometimes after many, many months of benefit. But we also know that some tumors in patients don't respond well to these drugs. So this idea of resistance to these therapies is really important, and figuring out what causes that resistance and then what do you do about it. The what next question that Terri mentioned is critically important. Beyond having these good drugs, we need to find out how to make them work as well as possible and better for longer. And so that means understanding the biology of how these tumors respond to RAS being blocked, and how does that create new vulnerabilities or weaknesses in those cells that we can then take advantage of in a 1-2 punch, RAS inhibitor plus something else. And how do we keep doing that as long as the patient needs it? So that's going to keep us all busy for many, many years to come. But that is being actively studied in laboratories, in the clinic, in pharmaceutical companies, really trying to understand, okay, now we have the foundation, which are RAS inhibitors, pill forms of therapies that will be generally well tolerated. What are the other building blocks we need to put on top of that to really make these drugs work better? And that's a tremendous challenge. It's a great challenge, a great opportunity, and something that all of us are working very hard on.
From a practical standpoint, if I have pancreatic cancer and I've been through 2 lines of treatment and I have progression, I'm still fighting it. How am I narrowing down the scope for what my next treatments are? Like in a today, not in a like we got to keep researching it perspective.
So is your question, how do you figure out what your next therapies
How
are?
do I know my next step? Let's
Yeah.
just say arbitrarily, I've been through chemo, I've been through surgery, and I've been through a clinical trial, pick one. And now I've acquired some resistance.
Yeah.
What am I looking for next? How do I know I've acquired resistance? I mean, is there another mutation that happens? What is it I'm looking for? What is the deciding factor for what's next?
Yeah, well, these are super important questions, and questions that you should not have to ask and answer yourself. And that by that I mean this is what your oncologist is for, and your oncology team, and they need to be up to date on current standards of care and treatment paradigms, but also up to date on what the options are available. And that may mean getting second opinions. That may mean having your oncologist be the quarterback of the team that says, you know what, I see some signs that the cancer's no longer responding to chemotherapy. Let's get you to this center where we can consider clinical trials. Or I'm going to get on the phone and talk to my colleague and we're going to discuss this. And, oncology is a team sport. And it's critically important that we all talk about the patients as much as we can. And, when we see patients at Dana-Farber, when patients are seen at any, multidisciplinary cancer center, it's a discussion around those patients. And what do we do? And let's put our heads together and figure out, are there clinical trials available? If there aren't trials that are open here, are they open elsewhere? Can we phone a friend and figure out, hey, do you have anything for this particular type of cancer? So for patients, I would say finding an oncologist and a team that you trust. Don't be afraid to seek second opinions when necessary, and do that through your oncologist and get their advice about second opinions that they should pursue. Organizations like KRAS Kickers or Pancreatic Cancer Action Network or the Lustgarten Foundation are phenomenal organizations to be able to connect patients to each other, but also connect patients to physicians and physician networks and trial finders. And so I'd say be resourceful, but don't do it alone. Communicate, collaborate, work with your care team, friends, advocacy organizations to find yourself in the best place possible.
Okay, so action item from what you said, don't be afraid of a second opinion. In fact, don't be afraid of a third opinion.
Yeah, absolutely. And I would say that as oncologists, physicians, we're not afraid of second, third opinions. In fact, we welcome it. And, it's about the patient and doing what's best for the patient. And that opinion might be, can you get your care closer to home or closer to a family network that, is supportive of you and helps you get through the treatments? Is there another trial available that maybe we don't have open, but someone else does? So, your doctors aren't afraid of those opinions. There's no ego in caring for cancer. And I really think, use all of your connections and resources available to ask your doctors for help. Ask your oncologist, should I be thinking about trials elsewhere? Can you recommend some to me? All important questions. And really, don't be bashful in asking for help, even from your doctors. Are we thinking about this the right way? And I think that's important.
Don't be afraid to use the T word, trial.
Yep. Yeah, I think exploring trials, making, and your doctors will help you understand, is this right for you? It might be the right marker in your tumor, but is the trial right? Does the back and forth to the clinic, does that work for you? Does that match up with where you are in your treatment course? Is the therapy likely to benefit you more than it is to cause toxicity? Those are all conversations with your doctor.
And definitely, I mean, we're not here to give specific information or advice about anybody's treatment, just kind of like help point you in the right direction so you know that when you ask the question, what kind of answer you're getting back and what it is you should be listening for. And so getting that information and asking about the trial and getting it, move forward with it, that's very empowering. It's very scary as a patient to ask for second opinion. And I know that you don't realize how scary it is, but it feels very threatening for us to go and do it. And yet at the same time, you have one body and your doctor can't die for you. He can't live for you or she. And it's at the same time, you have to be able to take that initiative and do something for yourself because time is of the essence when it comes to it. And now when I say time is of the essence, no doctor can help you on a Friday night at midnight. That's it. I mean, it's just not the way it's going to work, at least not when it comes to a new cancer diagnosis. So, all right. I've been delaying getting to some of the questions. I was trying to kind of like articulate them into natural conversation flow and have been struggling doing that. So instead, I'm just going to start spitting some of them out there at you. And so then we're going to jump around a little bit. Okay. So I guess this is kind of a lightning round. Are you ready for lightning round, I guess?
I am. Yep.
Okay. All right. So
We
some
need
of
all
these are
questions.
not going to be pancreatic related specific. And so we're just going to kind of go for it. All right. So when, see, so as an appendix cancer with G12D, okay, which is, we talk about it so much. So appendix cancer with a KRAS G12D, what kind of assessment, what kind of similarities does that have? To pancreatic cancer, are those like a trial situation, or it's like, just kind of outside your scope, can't pass on the answer?
Yeah, so appendix cancer, much more similar to colorectal cancer. And in fact, the genetics of appendiceal cancers or cancers of the appendix, very similar to colon cancers, less similar to pancreatic cancers. The role of RAS is important in each of those tumor types, pancreas and appendix cancer. But those types of cancers would more align with colorectal cancer clinical trials. And so the types of trials that are ongoing for colon cancer, we've learned from G12C inhibitors that combination of a G12C inhibitor with another therapy that blocks another protein, EGFR, that's approved in G12C mutant colorectal cancers. So there are some paradigms already established for what to combine RAS inhibitors with in cancers of the colon or appendix. But if you had a G12D mutant appendiceal cancer, you should seek out a gastrointestinal cancer specialist and consider trials for KRAS mutant appendiceal cancers or colorectal cancers, and it'd be a discussion around what type of trial options are available for those patients.
Okay, thank you. Next up, are you aware of any resistance occurring with KRAS wild type drugs?
Resistant, so by
Any
KRAS wild type,
resistance KRAS wild type drugs?
yeah. So the question brings up an important point that some drugs block wild type KRAS, or wild type means normal KRAS. So KRAS that doesn't have a mutation, but maybe is activated above by causing, RAS to go in the absence of a mutation. There are drugs that block that protein. In fact, that drug that we talked about, daraxonrasib, blocks normal or wild type KRAS as well as mutant KRAS. And so those trials are applicable for cancers driven by wild type or normal KRAS. And those can be driven by other alterations that signal to RAS and cause it to be in the on form. So there are trials out there that are testing drugs that block normal KRAS or wild type KRAS. And there are mechanisms of resistance that develop over time when cancers are treated with these drugs, irrespective of whether they're mutant RAS or normal wild type RAS. And so many of those resistance mechanisms are common, where they activate the RAS signaling pathway in some other way and bypass those drugs. So they exist. We're actively thinking about them and actively working to figure out ways to combat those resistance mechanisms.
Okay, so next steps for considerations if you're one of those people.
Well, next steps for considerations, if you had a mutation that caused resistance, for instance, would be to find a next generation drug that maybe still works when you have that mutation or blocks the RAS pathway in some other way, or uses other therapies that don't necessarily depend on RAS signaling. For instance, there are a whole host of other potential targeted therapies that block other proteins. There is a class of drugs called PRMT5 inhibitors that work in a specific setting, for instance, when you have a deletion of another gene called MTAP. And that mutation occurs at the same time in the same tumor in about 30% of pancreatic cancers. So there are other trials that you can think about, or other trials of therapies called antibodies, which are really therapies that target the surface of a cancer cell. So there are a whole host of other types of therapies, really an explosion of new therapies in oncology that we're trying to repurpose for pancreas cancers or GI cancers more generally. And those are other trials that if you had progression while you were taking a RAS inhibitor, one, is there another RAS inhibitor that you can give? If not, are there other therapies that would be applicable to those tumors, either alone or in combination, perhaps with a RAS inhibitor?
Yeah, okay. Thank you. All right, so next up. Let's see, no. So we have, there's just been, I'm actually just read this. Patients who've already had chemo and they've been on a RAS inhibitor like RMC 6236. What's the next step? I know you need to talk to your doctor. I know they're going to do more biomarker testing, and we're working on it. But like if you're just looking at the decision tree, and it's like at Dana-Farber, if you're seeing something in this line, you would be looking at these potential decisions, right? Are they going to be qualified for, you know, an ADC or some other trial or something that's going to work for sure? But if you're just generalizing the statement, what would you, what should a patient consider, or what would a patient expect to see happen?
Yeah, it's an important, and I would guess I would say a complicated question, in that it depends on what the patient has had before they received the RAS inhibitor. It depends on why the patient's tumor has stopped responding to a RAS inhibitor or started growing again. But generally speaking, the patient could think about, first of all, like you said, talk to your oncologist. But to see, are there other chemotherapy options, or are there next generation clinical trials that the patient could be eligible for? Because we now know that there are clinical trials to follow on prior clinical trials of KRAS inhibitors or other therapies. And as we mentioned earlier, there are a host of other new therapies besides RAS inhibitors that are exciting and promising in pancreatic cancer as well.
Well, and there's a lot of them that are specifically looking for somebody who has an acquired resistance mechanism as a result of being on an inhibitor. And so there are trials specific for that. It doesn't mean they're in every neighborhood, every place, but that is definitely one reason why you need to speak with your doctor about it to get it narrowed down. To that end, would a patient expect that they would get a new mutation testing to see if there's any sort of other genetic alterations that show up on their hit list that they might be looking at next? Is that kind of like one of the next steps that you would see? And then from there, be able to use that information to make decisions moving forward?
Yeah, that's right. And that's at, academic medical centers, that's very commonly done, where you can take even a blood sample and look for circulating bits of DNA from the tumor that you can sequence, and then ask, are there alterations that are emerging or mutations that are emerging when the tumor is progressing on the drug? And do those mutations point you in the right direction for a new therapy? So sequencing tumors, additional biopsies of the tumor that's growing, or blood sampling of the tumor that's growing, that can be a way that can point you to new potential therapies for patients. And so that's something that definitely talk to your doctor about. If you're in a place where that's not done commonly, it'd be reasonable to talk to your doctor about a referral to a center where that's done, even if to just go get a visit and an opinion. And as many people likely know, oncologists work all the time in teams. You can go get an opinion, go back to your close to home oncologist, but then that is a bridge between those two centers and those two doctors that they can work together and call on each other when necessary to help guide patient care.
Well, you can't talk about us if you haven't met us since we've shared our reports with you. I mean, let's just.
Yep. No, that's an important point.
Yeah. And so, I mean, so we have to, yeah, so we need to make that first step in order to be able to do that. And so, a lot of it, like I said, I personally had a big fear of doing it. So I think that's a lot of facing that fear factor, is being able to kind of move forward with it. So then you have a reason to discuss it. Okay. So you're not, when I come to you for a second opinion, you're not giving me the opinion that my doctor already gave me just because you guys are gonna, I don't know, go golfing together, right? You're actually looking at me differently.
Yeah, and that's what hopefully your original oncologist wants as well. Cancer is a humbling disease for patients, for physicians. So we all acknowledge our limitations in battling this disease and look for help, look for second opinions. We're constantly talking about patients at our own institution and constantly engaging with others and putting our heads together and thinking about how best to treat cancer. So when you go for a second opinion, you want an independent second opinion. And a lot of times that will differ from what your original opinion was. Your doctor also wants to hear that. They don't want to send you to see someone and hear exactly the same thing that they told you just because, that's what they told you. They want to know, is there something else that they should be thinking about? And many times it's eye-opening and a learning experience for the physician as well to think, oh, okay, that's how they do that a little bit differently there. And they learn from that. And then they think about that differently the next time. And it's not that anyone was wrong. They can both be right in two different paths to treat a patient. But what you want to do is you want to have all your cards on the table and then think through them together with your doctors.
Cancer is so much more strategic than we were kind of led to believe it was when I was like in high school, I guess. Or maybe
Yeah,
in the most
I
recent
think
movies. Okay, all the movies, it's like you go get chemo and ta-da, you're cured. Chemo and
no,
surgery, ta-da, you're cured or dead. And it's a lot more complicated than that.
yep, treating cancer is definitely requires a lot of planning and a lot of and honestly, we're fortunate 25 years ago, the first human genome was sequenced, and the first genomes of cancer tumors were sequenced 5, 10 years later. And ever after, we've had a roadmap and a blueprint to understand how cancer is different from normal cells. And that blueprint has given us a ton of things to think about. Why do cancers form in the first place? And now, what do we do about them? So we're constantly looking for that blueprint to plan and think strategically, to think about other drugs, to work with pharmaceutical companies to develop those drugs, to bring those to patients. So oncology is fascinating. And it's an amazing field to be in. But at the center of it is our desire to help patients and never lose sight of that.
Well, we appreciate that because we certainly need help. We're not out here doing it on our own. Okay. Next up is somebody had a genomic NGS test done initially on their pancreatic cancer tumor, and now the cancer has spread again. Would you suggest that they get a new genomic testing done? I think we kind of covered that, right?
Yeah, I think the answer is it depends. If the cancer's behaving in a different way or it's popped up on a therapy and growing differently than it was, then yes. Fortunately, we have what we often call liquid biopsies or blood-based sampling and the sequencing of that circulating DNA from tumors, circulating tumor DNA. That can be done without a repeat needle biopsy. But when necessary, we now know that these repeat needle biopsies of spots of cancer that are growing can be done quite safely and are well tolerated. And sometimes that's necessary to go back in, take another sample of the tumor and understand, well, why is it growing? When everything else is controlled, why is this spot growing? Or why all of a sudden did these drugs stop working? Sometimes that's necessary to go and get that additional information.
Yeah, and if you've been on a previous clinical trial, it might be information that could be pertinent as well, right?
Yep.
Okay. Can you discuss the various transport mechanisms, e.g. album and the Amplify Eli 002 trials, mRNA, et cetera, currently being investigated in clinical trials?
Yeah, I think this questioner is referring to uh, vaccine strategies now that are being used in pancreas cancer patients. And there are some really exciting results in the post-surgery setting, in the adjuvant setting. So the patient has a localized tumor, take the tumor out, and then you give a vaccine just like you think of a COVID vaccine or a flu vaccine, but this vaccine is geared towards the tumor. And the Amplify 201 study that they talked about, this is our peptide vaccines, are little tiny protein vaccines directed at KRAS itself. In another study, they're personalized vaccines, what are called mRNA vaccines, much like the COVID vaccine, where you figure out what markers the tumor is expressing. You design a vaccine directed at those markers, and you give that vaccine a personalized vaccine to those patients. And both of those two major buckets in studies have shown pretty dramatic results in the post-surgery setting, that the patients that had a response to the vaccine tended to do better. So that's highly encouraging, that not only could we have RAS inhibitors to shrink the tumor, surgery, chemotherapy, that we could add another tool in our toolbox to be able to fight the cancer, which is to ramp up the patient's immune system to go in and recognize the tumor. And the way to do that may be through vaccines or other strategies to really fire up the immune cells to go get the cancer.
Okay, great. All right, so next up, in lung cancer, there's immunotherapy, and that's kind of gauged on PD-L1 is one of the markers that's used. Is there something similar to that in pancreatic cancer?
Great question. In lung cancer, PD-L1 or PD-1 blockade is really standard of care in the frontline setting, the first line setting for lung cancers, including KRAS mutant cancers. And in fact, in lung cancer, much of the work of what do you combine a RAS inhibitor with is how do you combine it with immunotherapy? And how do you make that tolerable and combinable to be able to get that to patients early? In pancreas cancer, those immunotherapies, outside of a small subset of patients, have not really worked well. In fact, we just don't see the responses in pancreatic cancer to immune checkpoint blockade or anti-PD-1 or anti-PD-L1 therapies or immunotherapy. We don't see those responses to that single agent immunotherapy. But RAS inhibitors change the tumor in an important way. They cause an infiltration in the tumor of what are called T cells, or these immune cells, these infection fighting cells, but also potentially cancer fighting cells. So RAS inhibitors themselves change the tumor, make it more conducive to immune cells that come in. And so I think there's reason for hope that that changed tumor now, that with RAS block, shutting down all those negative signals that come from RAS that keep the immune cells away, when you shut down RAS, immune cells may come in and they may be active against the tumor with the right immunotherapy, whether it's a vaccine or this immune checkpoint blockade. So those studies need to be done, but I'm optimistic, and not only optimistic, but I think it's going to be necessary to figure out a way to turn the immune system against the tumor to get really long-term, durable remissions from pancreatic cancer. We know we have RAS inhibitors, we have chemotherapy. We need the immune system to come in and clean it up and keep it at bay. And so that's something we're all working very hard on.
Are there trials in that line right now?
There are trials in planning. The drugs are currently at a point in development in pancreas cancer that they're not yet there where we're combining them with immunotherapy actively, but that's in everybody's mind. That's where we need to go, and we need to do that experiment in preclinical models in the lab, but also ultimately do that trial in patients.
Okay. Well, it makes me wonder, is there something besides like the PD-L1 that might be an indicator of who would benefit from it?
Great question. The PD-L1 is a molecule or a marker that's expressed on cancer cells that kind of keeps the immune cells at bay. These drugs that come in and block that unleash that blocking that interaction, that negative interaction, unleashes the immune cells. Those are called immune checkpoints, almost like a checkpoint at the border, where you go through and you're blocked by the border patrol until you get clearance to go through that checkpoint. I'm like, okay, is this tumor cell going to get eaten by the T cell or getting zapped by the T cell? If that checkpoint's in place, it may stop it from getting zapped. In addition to PD-L1, there are other immune checkpoints that can get expressed on the surface of cancer cells and T cells, and those interactions can regulate how those immune cells, those infection or tumor fighting cells work. There are other types of immune checkpoints besides PD-L1 that can be expressed on cancer cells. And in pancreas cancer, there's some data that suggests it isn't PD-L1, but it's other checkpoints that we need to be blocking. So, and there are antibodies and therapies that block those other checkpoints. So that's an important area of study, both in the lab and in the clinic. Which of those checkpoints is most important to block to unleash the activity of these immune cells against cancer? And we have to get that right. We have to figure out what those checkpoints are, how do we block them, and how do we combine them with RAS inhibitors.
So get to work.
Yeah, tall order. We're trying.
I know you are. Okay. Let's see what else we have in here. Right, I did that. Are there any trials for KRAS plus another mutation, like a combo, KRAS plus GNAS? Let's start there.
Yeah, good question. GNAS is another protein like KRAS that can be a driver of cancer, and it's when it's mutated or changed, it's also in the on position. So currently we don't have drugs for GNAS, altered GNAS proteins, mutant GNAS proteins. There's some interesting work in the laboratory from a number of labs looking at signaling pathways downstream of GNAS that are activated, and how do you manipulate those signaling pathways to take advantage of that activity that could, ultimately kill tumor cells. But we don't have those drugs yet. But as I had mentioned, the PRMT5 inhibitors in tumors that have a deletion of a gene called MTAP, those mutations and those vulnerabilities co-occur in about 30% of pancreas cancers. There are now clinical trials actively being planned to combine RAS inhibitors with PRMT5 inhibitors. And at the same time, in the same patient, again, this 1-2 punch that we're going to take advantage of these two vulnerabilities at the same time and combine those therapies to hopefully really wall up the tumor cells and give them two effective therapies at once. So we're constantly looking for those types of vulnerabilities, including new vulnerabilities that might be induced by RAS inhibition or that co-occur with RAS mutations, and trying to layer on those different therapies.
Okay. All right. And all right, I missed a whole section of questions here. So again, a little out of order. What KRAS drugs are currently available by prescription? And are there any firm dates for the new ones coming online soon?
Yeah, the KRAS inhibitors that are available by prescription, meaning those that are perhaps FDA approved, include 2 G12C inhibitors in lung cancer for use in lung cancer, sotorasib and adagrasib, and then those 2 KRAS inhibitors combined with antibodies that block the EGFR protein in colorectal cancer. So single agent approvals in lung cancer, combination approvals in colorectal cancer, and both adagrasib and sotorasib, 2 KRAS G12C inhibitors. There are not approvals yet for pancreatic cancer. There are many other KRAS G12C inhibitors in clinical trials that look promising and down the line are likely to get approvals. There are many KRAS G12D, pan-RAS inhibitors, pan-KRAS inhibitors in active clinical trials in pancreas cancer that hopefully down the line we may see approvals in those with those drugs as well.
And the G12C treatments, I think there might be a smattering of approvals for like GI cancers that have KRAS G12C or something that has like a specific mutation.
Yeah, there are some indications and guidelines
Yeah.
to consider those. But in terms of major approvals, lung and colorectal cancer.
Yeah. Okay, we're coming up quick, I hope, right?
I hope so. Yes.
Okay, and then let's see oops, I did that one. And we're getting close to time, so I'm looking through here, see if there's anything like, okay. Yes, clinical trials are really hard to find, not going to lie about that, no matter what. Well, that's a good question. I'll ask you this one, and then we'll wrap up with one more wrap-up question. Okay, given what we know about targeted therapy, Why is chemotherapy still a first line in most cancers?
Yeah, good question. Well, first of all, I'll say, chemotherapy does work. It gets a bad rap in large part because of side effects and toxicity, but it is effective in a number of patients, including in pancreatic cancer. So starting with that chemotherapy, when tolerated by patients, does have an important benefit to patients. Targeted therapy depends on, as Terri said earlier, having the right biomarker. And that's true in, say, EGFR mutant lung cancer, BRAF mutant colorectal cancer, KRAS mutant lung, pancreas, colorectal cancer. So you have to have that right biomarker. In cancers where that biomarker is present, for instance, EGFR mutant lung cancer, the standard of care is to give the EGFR inhibitor in the first line. That said, it's also been shown that giving the EGFR inhibitor with chemotherapy in the frontline setting works better than getting the EGFR inhibitor alone. So that combination of drugs is effective. In terms of why we give so many patients chemotherapy up front, oftentimes it's because those biomarker relationships or those targeted therapy opportunities don't exist in those patients. I will say, with the development of RAS inhibitors, the number of patients that have opportunities for targeted therapy goes way up because RAS is so common. It's really, drives so many different human tumors. So good RAS inhibitors that go through the right clinical trials is going to dramatically increase and improve the number of patients that can get a targeted therapy. But I still predict that we're going to be combining those in some way with chemotherapy, other targeted therapies, or immunotherapy. And so these tumors are so complicated that we know that we need combination therapies to really knock them back and keep them at bay.
So even though chemotherapy sucks, and that's personal experience, it does do the quick shock treatment to your body to knock out a lot of the cancer that could potentially be growing. And so as much as I want to, say from, a Monday morning quarterback share, like, I think chemo's going to go away overnight. Just from watching what's happening, I can't see, because there still seems like you're going to need some level of that, right? It needs to be used appropriately, but
Yeah. And I would hope that one obvious way that we can make advances early on is if we have a good RAS inhibitor, a good targeted therapy, maybe we can dial that chemotherapy back to where it's effective but doesn't have so much toxicity. So instead of giving everything but the kitchen sink, dial it back to 2 agents instead of 3 with a RAS inhibitor. And so I'm hopeful that, more modest chemotherapy may still have a benefit with the RAS inhibitor in certain cases. So, but I agree. I think
I want to say that. I want to say that, but just from, like I said, just armchair like watching, I think that it's going to need to have some version of that. Maybe not everybody needs it, but we'll need to stick around for a while, and the timing is right. As a patient is kind of like going through, and let's just say between hearing from you talking, they go through, they do a little armchair, quarterbacking, looking for clinical trials, and they're coming in. How can they best know what to prioritize their conversation with their doctor? I know we're not oncologists, and I know we don't really want to be one, but we do want to feel a sense of confidence when we're going in and speaking to the doctor. And that's like what our org and many of them are about, is having a sense of empowerment and comfort in going in and speaking. But I want to use your time wisely, okay? And so if I'm coming in and I'm like, I can't just, nobody can drink from a water hose. How is it I'm going to go through and say, okay, well, these are the things I would like you to consider for me, doctor. Where do you suggest I go? Maybe Farber's my next best choice, because you have this list of trials would be best considered. How do I gauge that conversation? Because I can't, you just don't have time to be traveling to 5 different places.
Yeah, I think getting to a major, assuming oncology care can be delivered well at many, many places in the country. I think we have a wonderful oncology system, world-class oncology care in the United States. But when it comes to clinical trials, getting to these, centers of excellence, National Cancer Institute designated comprehensive cancer centers where there's a mission and a focus on not only, clinical care, but clinical trials, patient education and outreach, that can be a really good first step, or getting to one of these centers that focuses on clinical trials. And then getting to those doctors who can break this down. It's hard for all of us to keep this all straight but we do it together in teamwork and lots of meetings and paying attention to the literature, and we keep each other abreast of what's going on. So getting that second opinion or that referral, if needed, to these centers can be the gateway to more information and the gateway to the counseling and advice you need to make your decisions. And that counseling and advice can be for you, can be for your oncologist and physician. And so that's the most important thing I could recommend, is get to the right place. Use, resources and KRAS Kickers and others to point you in the right direction, not necessarily to get treatment advice, but to point you in the right direction, experts or centers or trials that may be right for you to consider, or the advice you need to get. There's so much on the internet, so much on social media. You can't go to sources that you can trust, and go to physicians and get your advice from your doctors that you trust.
Well, thank you. I think that almost does better than what I was planning on ending with. So I was like, hey, so do you think we're going to be curing this over the next 10 years? And it's like, I can fill in the blank with the answer. The answer is, well, I really hope that we can, and things are moving in the right direction. But the truth of the matter is that, we need something right now today. You all know that. Okay. I mean, as oncologists, as researchers, okay, it's, that. I see that. I hear that. I got to say, and this is a compliment to you and to the industry overall, is the way that you're coming at this as being able to how to attack these cancers, and coming at it and coming with new approaches for logical, systemic ways of getting in front of the cancer has been just so, I don't know, kind of renewed my spirit in cancer oncology, right? When I lost my mom when I was young, and I thought you saw the oncologist, then mortician. Turned out there's this huge step in the middle, and it's because of people like you that you do the research, and you actually have a sense of, let's keep you going, let's keep you alive, and let's give you some life through this. And so with that, I just love to thank you for joining us today and being able to kind of answer a bunch of these questions and sharing your insights around it all. So thank you, Dr. Aguirre, for being here. And I'm sure I'll see you at one of the upcoming conferences again in the very near future. Or maybe, I don't know, maybe at some point I'll get to Boston again.
Yeah, well, thank you for the opportunity and the privilege of speaking here today. And thank you also for all that you do for patients.
Well, we're all in it together to kick cancer's KRAS, right?
I agree.
Okay, great. All right. Well, thank you all for this. And we'll have a webinar recording of it that'll go out in the very near future. Okay. Have a great rest of your day. Thank you. Bye.
Thank you. Take care. Bye.